Good morning, good afternoon, and good evening. Thank you for joining us for Advanced Blast Freezing Technology for Drug Product Development. I'm Lauren Berry, your event moderator and executive assistant for Train Technologies Life Science Solutions business. All attendees have been placed in listen-only mode. This presentation will be recorded and is best viewed using GoToWebinar software for PC or Mac, or by using the GoToWebinar app on a smartphone or tablet. If you have technical difficulties, please send a message in the chat window and we'll attempt to troubleshoot your issue. During this presentation, you can ask our subject matter expert questions using the question window in GoToWebinar. Once this presentation has concluded, our speakers will answer your questions live in this session. Let me share a little background on our presenters today. I'm joined by our speakers, Scott Masella and Rob Shido. Scott manages the life sciences portfolio of products and solutions for Train Technologies, including FARRAR freezers and storage freezers, walk-in cold rooms, and integration of core technology into life sciences. With more than 20 years of experience while holding positions of progressively increasing responsibility in engineering and product management, Scott has gained a deep understanding of how to apply innovative technologies, techniques, and products to solve key customer problems. Recognizing the challenge of adopting sustainable practices and tools without compromising quality can be daunting. He has collaborated with numerous teams to provide cutting-edge solutions that contribute to achieving their sustainability goals. Scott holds a bachelor's in mechanical engineering from Tufts University in Boston, Massachusetts. Joining Scott today is Rob Shido. Rob is a senior systems engineering manager at Integris. Rob holds 2 degrees from Cornell University in Ithaca, New York, and has spent more than 20 years bringing products from R&D to production in a variety of high-tech industries. Currently acting as a senior systems engineering manager in the Integris Billerica Life Sciences divisions, his team is focused on implementing cold chain solutions for biopharma customers across a variety of market segments and customized solutions for cold room applications, leveraging his years of experience working on complex projects. I will now turn it over to Scott to begin our presentation.
Thank you, Lauren. And hello, everyone, and thanks for attending. Today, we're going to discuss ways to improve your freezing processes, including tools and equipment. But before we do, we'll get you up to speed with our companies, Integris and FARRAR. Rob and I will discuss the common challenges with respect to the freeze and thaw processes in your biologic drug process. We'll then take you through the components of our integrated solutions and dive into the data gathered from the process testing results and how it supports solving the common challenges in this presentation. Finally, we'll talk through our unique service support offerings that will allow you to experience these solutions in or out of your facility. So let's get started. We'll start this presentation giving you a little bit of information around our companies to familiarize yourself. I'll let Rob get started and talk about Integris first, and then I'll follow up with FARRAR. Go ahead, Rob.
Greetings, everyone. This is Rob Shido. Thanks to Lauren and Scott for the introductions. We'll briefly cover a little bit of background on Integris. So, Integris was founded in 1966, now more than 50 years old as a company, and focuses specifically on materials and process solutions for a variety of high-tech industries, including semiconductor and life sciences. Our headquarters is here in Billerica, Massachusetts. That's about 30 minutes north of Boston, if there's no traffic. And we currently have about 9,400 employees across the globe that are divided up into 4 separate business units. Those are our Advanced Material Handling, or AMH group, Microcontamination Controls, MC, Specialty Chemicals and Engineering Materials, SCEM, and Advanced Planarization Solutions, or APS. Through a series of acquisitions over the years, Integris has grown. And in the last couple of years, through market growth and additional acquisitions, we currently have a revenue of about almost $4 billion per year. So Integris is primarily known as a semiconductor company, but we founded the life sciences division to transfer some of the knowledge about purity and yield that was learned in semiconductor over to materials and processes that can apply to biopharma. You'll find that we represent fields including fluid handling, bioreactors and mixing, single-use assemblies, filtration and purification, clean chemical storage, particulate testing, and also specialty chemicals that are used by a number of different processes inside of biopharma. So in terms of portfolio and what does Integris bring to the table in life sciences, we'll start with our Aramis cryo assemblies. Those consist of bags, connectors, interconnects, manifolds, and protective shells that can be used in cold temperature applications. Those are the ones that we're primarily going to focus on today during the presentation. We do also have single-use liquid and powder systems. Those can be polyethylene materials, manifolds, assemblies, and accessories for rocker assemblies and mixing. In filtration, we've got a number of different materials that can be used for contamination control or filtration of liquid species. And also we have technology in process analytics, specifically particle size analyzers. So now we'll transition to Scott for a little bit of information on Train.
Thanks, Rob. For those of you not familiar with our parent organization, Train Technologies is a large publicly traded company, and our stock ticker is TT. Our brands include Train commercial and residential HVAC equipment, Thermo King refrigerated transport equipment, and our newest life sciences applications brands, FARRAR, Helmer Scientific, and Diversified Laboratory Repair. In total, we represent 38 manufacturing locations worldwide, about 37,000 employees, and more than $16 billion in sales. So Train Technologies has identified 4 key markets to attack sustainability through heating and cooling. Life sciences are our latest solutions market. Train has served this market through commercial HVAC and transport refrigeration for many years. The same decarbonization effort going in on our HVAC side can be applied to life sciences cold storage needs as well, one of the single largest market contributors to greenhouse gas emissions. Life Science Solutions, like I said, is our newest business focus for Train. We're committed to working with customers to solve the impossible. So we leverage Train Technologies' decades of expertise in precision temperature management to preserve the stability and viability of critical materials in health and life sciences. From powering groundbreaking medical discoveries to safeguarding essential vaccines and medications, our solutions meaningfully address and advance the well-being of people everywhere. So Train Technologies Life Science Solutions is our industry's leading expert in precision temperature management. Our portfolio of solutions, which include FARRAR, which we'll talk about today, Helmer Scientific, based in Indianapolis, and focusing on the clinical market segment, and Diversified Laboratory Repair, based in Maryland, which focuses on repairing and servicing lab equipment and other service processes, a dedicated hands-on service every step of the way. So we invest in innovative, sustainable engineering that solves your problems before they arise, enabling peace of mind and long-term success for your business. So now that you have a good understanding of what our businesses are and how we got into the life science space, let's take a look at more of the wide variety of offerings inside and outside our life science market sector. And let's dig more into the freeze and thaw process and challenges that keep you up at night. We've got a lot to cover, so let's get started. I'll turn it over to Rob to take you through a study his team executed recently.
Thanks, Scott. So before we start talking about the future of freeze and thaw, let's cover a little bit about the history and how we got to where we are today. So in the early days of bulk drug substance freeze thaw, there were really 2 challenges that customers were looking to solve. The first is that generally drug manufacturing processes are decentralized. That means that the location where the drug is physically manufactured is not always the place that that drug is put through final fill or into final container and ultimately dispensed to patient or to the point of final use. The second is that many drug substances today are temperature sensitive. They have limited lifespan at room temperature or chilled temperatures, which requires them to be shipped in a frozen state where they can be preserved for a longer period of time or retain their efficacy over a longer period of time. So initially, those drug products were frozen in open-top, aseptically filled bottles in general storage freezers. So these could be lab freezers, generally referred to as cold wall, using bottles or other containers that were generally available but could be transitioned to use in the process. So if we look into today, there is more of a focus on closed single-use systems, so less use of reusable or stainless steel equipment in the market, more on that single-use system, but a significant focus on process validation of each of the steps of the cold chain process to ensure consistent drug product quality at any point in time. As we look forward, there's much focus on controlled rate freezing, commonly seen in plate freezers, but now also available in blast freezers through the assistance of FARRAR. And we're going to be talking about advances in full closed system processing. So as Scott said, we did a study last year to look at some of the market around the freeze-thaw space and in a number of different areas of the biopharma industry. So one of the focuses specifically was looking at bottles. So when we talked with customers with bottles and we asked specifically about pain points, this is a summary of the results that they had given us. So at first glance, it looks like contamination at 28% is the primary pain point associated with that particular type of consumable. However, if we look at the rest of the chart, you'll see the tabs around slow freeze, uneven freezing, freezer space, slow thaw, and uneven thaw. Those actually add up to a higher percentage than the contamination that is shown in the right-hand side, commonly represented by filling or draining using open-top containers. So what that says is it's the freeze-thaw process that actually causes some pain for our customers, not just the contamination. Next level of concern was custom caps and connectors. How do they assemble these bottles in their process? How do they connect them, and how does that integrate with the rest of the system? So as part of the transition to bags, we wanted to understand a little bit about what customers currently use. The wide majority of the customers that were surveyed currently have processes that consist of bags and bottles. That represented about 78% of those people that were surveyed in the process. About 11% only used bags, and 11% also used bags, bottles, and cryo vessels, so a combination of different temperature ranges inside their freezing systems. Surprising to us was that 0% of those surveyed only used bottles in their process. So if we take that same data and we transition to asking those customers about what they see that they would be using in the future, 60%, a good majority of those, see an increase in the use of bags in the spaces that they currently have in their manufacturing processes, while only about 40% say that it's going to remain approximately the same. So to graphically place this a little bit here, on the left-hand side, you've got what you might call first wave, open-top containers, lab environment, cold wall freezers without controlled rate freezing inside of them. Today, we've moved in to have bags taking more of the space inside of that market, but definitely single-use consumables as a whole being more than it was several years ago. But a really big focus on that process control portion of it. So as we look through each of those steps and how the drug product goes from room temperature to a cold temperature and back again, what controls can be put in place to enhance the quality. And then as we move forward and start talking more about controlled rate freezing, commonly associated with plate freezers, we now have solutions using the Aramis bag assemblies, blast freezing shells, and the controlled rate chambers from FARRAR to transition to controlled rate blast freezing. So where are we now? We've got slides that show that there's an uptick in the use of certainly single-use systems and bags, move towards controlled rate freezing, but how will we remedy this for your process products and patients? How can we address the solutions of each of you? We're going to talk about some of those solutions. Before we get started directly into those solutions, let's just take a quick review of what we're going to refer to as the cold chain workflow for the rest of the presentation. So this is the workflow that really happens after the drug product has been manufactured, whether it's purified, diluted, or concentrated. it needs to go through a process that it is put into a secondary container to be moving through a transportation process. First step of that process would be fill. So that is transferring that material from its initial container into usually a series of smaller containers that are easy to handle and transport or distribute to multiple facilities. Generally at that point, those bags are also protected, meaning that they're put inside of an enclosure to protect them from physical damage during the transportation process and to be robust during freeze and thaw. Obviously, next comes freeze. We're lowering the temperature of that drug product. Some of the data you'll see here will be around processes that need to cross below -60°C. They're generally brought to -80 to ensure that the drug product remains below -60. Following that freezing process, the drug product is stored until it is needed for transport. So that could be in the facility that it was manufactured in. It would then be transferred and shipped using a variety of different methods to relocate that drug product to a different physical location, at which point it is then thawed, returned back to room temperature, transferred potentially into a different vessel, and ultimately dispensed in whatever the final use may be for that. So in the case of a vaccine, that may be the little glass container that you would find in your doctor's office right before they would put it into a needle to inject it as part of the vaccine process. So shown here are some of the examples of the products that Integris and Train can offer in that space, from Aramis bags and assemblies, manifolds, shells, to freezing and to process freezing and storage freezing solutions offered by the combination of our two companies. So now we're going to talk about how to apply those Integris and Train technologies to this workflow.
So we can summarize the most common workflow challenges into 3 buckets: financial, process and product, and finally sustainability. I'll cover the challenges here, and as we go through the presentation, we'll systematically answer each one of these, and in the end we'll kind of summarize. So the top financial challenges include upfront capital investment. So control rate freezers can cost over $1 million, not including the infrastructure, which may include a chiller for water cooling the compressor, the electrical hookups, the floor space costs, and etc., to house and operate this equipment. Coupled with the investment is the concern that the equipment will not meet the process needs. So when that happens, users often change the process to best fit the equipment. The cost to maintain this equipment is also pretty high as well, with specialized freeze and thaw equipment. Reliability and uses other than the design intent further reduce the reliability, causing unplanned downtime in the manufacturing process. Floor space and ceiling heights are often factors as well and must be considered when operating control rate freezers, while also accounting for other infrastructure requirements like chilled water, HVAC loads, and liquid nitrogen. Finally, on the personnel side, specialized training and know-how is definitely required to work the equipment and also troubleshoot it. The human-machine interface may be complicated as well for an operator, and replicating this process in other factories takes time and training effort. So on the process and product challenges, they all begin with the vessel. So bottles are open systems and can introduce contamination. Ensuring you fill and cap the bottle without contamination is sometimes a real headache and requires specialized equipment, processes, and environments. Bottles can also take a long time to freeze and have more inconsistent freezes, meaning the center of the bottle will freeze significantly slower than the outside of the bottle. And in addition, using improper equipment to freeze those bottles or bags will dramatically increase freezing time and unreliability and results of the process. So inconsistent freeze rate within the bottle can cause cryo concentrations, which ultimately damage your product and result in product loss or yield losses. So bottles do not efficiently use up the space within a freezer either. Those round shapes and angles don't occupy space as effectively as a rectangular shape might, and more often than not, you can't stack them either. You need a shelf for every layer of material. So particulate, leachable, extractable profiles for those bottles are often not favorable. As with any vessel, cleanliness and the ability to be inert with the material inside is critical. So the top sustainability concerns include some control rate freezers use oil as a coolant within the plates. These oils are harmful to the environment and can be harmful to human health if breached from their contaminant structure or containment structures and systems within the freezer. Single-use systems are also synonymous with not being recyclable. So multi-layer films cannot be easily disposed of because you can't separate the layers post-use. In addition, many of these materials often are required to be decontaminated before recycling, making recycling expensive and complicated. So now we're going to get into the integrated solutions. So our solution contains 3 key elements: a bag, blast shells, which you can see here inside the freezer, and the control rate chamber, which is surrounding around the shells. We'll go into details with each of the components and how they work together to give you a complete solution that solves many, and by many I mean all, of the challenges we talked about just previously. So I'll let Rob get started describing and detailing the bags and blast shells first, and how they solve the key challenges associated with storage vessels.
Great, so thanks for the overview, Scott. And as you could see from the previous picture, one of the more interesting things inside of there is the polymer blast shells. So I'm going to spend a few minutes talking a little bit about some of the features of this new technology that's currently available on the market and how that can relate to the processes that we've been discussing today. So one of the big features that we try to incorporate into these blast shells is actually reusability. So as compared to other single-use containment solutions, these blast shells have been validated to be reusable. And during that validation process, we have tested them for 3 complete cycles. That's key because on the left-hand side of the picture, you can see how they're assembled, yet due to the design of the shells, they can actually be folded into the half shells on the right-hand side and stacked, meaning that they take up reduced space during shipping if they're transported between different facilities. Robustness has been tested on these at cold temperatures and at room temperature using a series of operational tests, as well as what we like to call foreseeable misuse. So that would be drop tests and slide testing to ensure that the drug product remains intact inside of the Aramis bag, inside the shell, during all sorts of different types of handling that could occur. Another key area was around ergonomics of using these types of pieces of equipment inside of the freeze-thaw workflows. So not only do you have employees in the facilities that are going to be working with clean room gloves, you also have employees that are going to be working with cryogenic gloves, because if these shells are transported at those temperatures of -60 to -80 C, you need to have protective equipment in place to be able to move those shells between. So there's a number of different handholds designed to accommodate a number of different sized hands to be able to safely move those shells between places inside the facility. Another key differentiator of this product in the market is the way that the bags can be filled. There's actually 3 separate methods. So depending on the customer's process, they can use any of those 3. The bags can be filled and then put into the shells. So in other words, filled in advance of being protected. The bags can be placed on a half shell where the shell is used as a tray to move the bag through the facility. It can be filled in that state. And the third possibility is that the bag can be installed into the shell fully assembled, and we have access to the tubing through the side of the shell so that the bag can be filled when it is fully assembled inside of that shell. Last key feature to discuss here is just going to be that there is a defined space for tubing and large connectors to be located inside of those shells, knowing that each of our customers may have a slightly different size tubing length or need for connectorization. So moving into the shell at this point, we'll talk a little bit about some of the Aramis bags and their differentiation in the market. So one of the biggest is that the Aramis bags are known for being extremely clean. They are assembled in a Class 5 clean room, which is one of the highest quality ratings that involves a lot of air exchange and making sure that particulate is evacuated from that space. Our other assemblies and the non-fluid contact portions are still assembled in Class 7s. So if we look at some of the testing data around those Aramis bags, we've got an example here that shows a comparison to some common EVA bags that are on the market. Chart on the right is showing the relationship to USP 788 and the number of particulates that that particular standard allows to be in place based on the size range of the particulate. So in the larger range from that 25 to 400 micron size, standard is 3 particles in that space. You can see here there is no gray bar over there that represents the Integris product line as compared to the competitive products that are in blue and orange. That means with 24 replicates of that test that 0 particles were found in that 25 to 400 micron range. Chart on the left shows the 10 to 400 micron range where the limit is 25. Integris very far below the competitive products in that space. As you can see, the standard deviations of some of those products would take it above the limit for that USP 788, which means that overall Integris assemblies have much lower particulates than their EVA competitors. Another chart here breaking it down into more different particulate size ranges. Again, Integris staying far below the competitors in terms of the total particulates that are inside of those tests. Of course, cleanliness is one of the things that Averamus has over its competitive solutions. Another is robustness, its ability to stay in its shape and retain integrity during low temperature or high temperature process steps. Of course, additional information here is always available in the Aramis validation guides or through our application support teams. Back to Scott for some information on some controlled rate chambers.
Thanks, Rob. Those blast shells are really unique and really are designed to support freezing without plate freezers. So let's take a closer look at a control rate chamber that accommodates those blast shells very effectively. The left picture shows the blast shells oriented in the chamber between our ductwork on the left and right. And the picture on the right is our control rate chamber LC model with the touchscreen HMI. This model has an easy profile programmer, ability to view the freeze process with a temperature graph, and also the ability to add a second probe for temperature tracking at the bag or bottle level. I'll explain the core system here on the following slides. So I wanted to introduce the model 4000 control rate chamber from FARRAR and give a short overview on it. When you're freezing materials, even small batches, it's important that you are using equipment with sufficient cooling capacity. You might ask why. With 2 easy reasons. One, you want it frozen fairly fast. Your yields and processes probably cannot afford to wait for a slow freezing process, typically found when you're using a ULT. And 2, putting excess heat load into the chamber will overwork a compressor set and leading to premature failures if it's not designed to handle that extra load, even for the best high quality units on the market. The refrigeration system inside this control rate chamber boasts 3 key features that make it one of the fastest, if not the fastest, mechanical blast freezer on the market. Number 1, the high output compressor set. So we have 3.7 kilowatt and 2.6 kilowatt hour kilowatt compressors. The best ULTs on the market have just 750 watts or less in each of their compressors. And unlike plate freezers, the control rate chamber can be air cooled, so you don't need to have a chilled water line, and doesn't circulate hazardous silicon oil. So number 2, forced air convection. So you've heard us talk about forced air convection in other webinars. With a complete chamber air exchange every second, it's removing heat from the material at a much more than double the efficiency over natural convection. This is no different than your convection oven at home cutting your cooking time down by 30% or more. And finally, the electronic expansion valve meters the refrigerant, so when you do not need all that cooling capacity, such as when you're ramping down and holding at a specific temperature, the unit can quickly reduce the amount of refrigerant to control the freezing rate and then expand it again when it's needed. ULTs typically use a fixed cap tube, which is dialed in to be most efficient at -70 or -80 degrees and the amount of refrigerant needed to maintain that steady state and account for an occasional door opening. So what this all does is it produces tight uniformity within the chamber. And in an empty chamber with running, it's about 0.1 degrees Celsius anywhere in the chamber. When you add a significant heat load, such as a load of warm product that it may ramp up to about plus or minus 2 degrees, especially around that warm product as it's cooling, and down to minus 80. So the unit has 3 built-in profiles ready to go, a thaw profile, a blast freezing profile down to minus 80, and a freezing profile down to minus 40. But you can add your own custom processes. It's got integrated thawing, as I talked about, blending a hot gas heater with the forced air convection, quickly warming product up to minus 40 degrees or anything cooler than that. I want to take a quick note and kind of point out one area on the screen. You'll see the airflow. So you can see quickly our ducting is moving from the left to the right, moving that cold air across the bottles in this picture. And whether you have bottles or bags, it's exactly what the airflow is doing. And it's moving and circulating back around through the evaporator, removing the heat, and then back again. It's doing that once a second. So now we'll take a more deeper look at the refrigeration design. So before we can really appreciate the speed of freezing, we have to take a basic understanding of the thermodynamics behind the design. I'm with most of you. I think probably try to simplify this as much as possible, but we'll focus on the heat loads of the Model 4000 and against a typical ULT and their ability to remove heat. So the Model 4000 has a standard and a standard 600 box ULT are very, very similar in terms of their cabinet dimensions. So rather than getting into the details on how we calculated it, just we can assume that they have very similar heat loads coming in from the ambient environment through the walls, through the door, and through the door seal. And that can be measured about 158 watts. As a forced air convection freezer, the Model 4000 also has to consider the additional heat source from our blower. And that blower, we know from our design, contributes another 325 watts of additional heat, bringing the total heat load inside our rate chamber to 484 watts. On the FARRAR 4000 series, our refrigeration system is also designed to remove 2100 watts of heat from the system. That's what you see there on that Q design. A typical ULT's Q design is typically designed to remove between 200 and 250 watts of heat. So let's just call it 250 watts and assume it's a beast of a ULT. This leaves about 92 watts of available cooling capacity for door openings and freezing warm product. Not a ton, and explains why door recoveries and freezing materials can take longer than you probably would like. For that Model 4000 rate chamber, that 2100 watts of our design minus the 484 watts leaves us with 1616 watts of available energy to remove heat. And that's 1524 watts more than a ULT, 17 times more, all devoted to removing heat from products and samples inside. It's a key reason why this isn't specifically designed just to do this, freeze product. So what's that cooling capacity really mean? What's it really feel like inside your process? So if we compare the performance versus a footprint, I think the advantages really stand out. I'm going to use a study that was performed by Integris, and Rob will detail about it a bit later in the presentation, along with a 2-liter bag study. So in this particular case on this slide, we're looking at 5-liter bags filled and placed in 3 different ULT freezer brands, and also the FARRAR control rate Model 4000. The ULTs each could hold about 50 liters or 10 bags, and the Model 4000 holds 9 bags or 45 liters. So the average freeze time for the bags in those ULTs was 71 hours, almost 3 days, and in the Model 4000, 11 hours. So this Model 4000, what that means is that Model 4000 can make 6 runs and be halfway through the 7th run when the ULT finally finishes its first run. This equates to about 270 liters of freezing capacity in roughly 3 days versus 50 when using ULT. So to be equivalent, you'd have to have 6 ULTs that take up roughly 197 square feet or 18 square meters of floor space compared to a single Model 4000, which would be about 52 square feet or 4.9 square meters. That's almost 4 times more floor space needed for the ULTs to freeze the same amount of material. So this really accents and shows how the Model 4000's cooling capacity and forced air convection really help freezing in these use cases. But Rob, like I said, Rob will be showing you just in a few minutes how the control rate freezer freezes bags. But before he does, I wanted to take some time and do a quick deep dive at how the rate freezer does with bottles. So since we looked earlier in the presentation around the majority of users have a mix of bottles and bags in your facility, it would be a good idea to show you just how the unit also performs with bottles. So in this slide, we're looking at 3 sizes of bottles we looked at: 2 liters, 4 liters, and 10 liters. We added temp probes to each bottle in the center to determine the internal bottle temperature, so we can verify when it achieved the temperatures we've got here, in this case, minus 60 degrees C. And you can see this temperature probe design in that carboy picture at the very bottom. The key takeaways here are this: we can fit 180 liters or 90 bottles into the 92 liter bottles into the chamber. The average time to freeze from ambient, that's 20 degrees C, down to minus 60 took about 8.8 to 10 hours. So the fastest bottle was 8.8, the slowest bottle took 10. The picture to the left for this one is in the middle. So it's a left picture on the middle of the page. We also fit 24 4-liter bottles, or just over 86 liters, into the chamber and froze those bottles to minus 60 between 4.4 and 7.4 hours. And finally, we took 12 10-liter carboys totaling over 112 liters and froze them to minus 60 between 7.4 and 11 hours. These results also tell a slightly different story. It's really important to choose your storage vessel with care. So not having a high surface area vessel and material with poor thermal conductivity heavily contribute to freeze and thaw speed. You can see here the 1-liter bottles, even with 68 more liters, can freeze faster than the 10-liter carboys. Just a surface area available to cool. So I'm going to turn it back over to Rob to talk about, to take you through Entegris's unique ROI calculator that helps you pick the ideal freeze and thaw solutions for your process, and then followed by a deep dive into the bag freeze and thaw data.
Yep, so if transition from one manufacturing technique, let's say bottles to bags, is of interest to a customer, or evaluating how a controlled rate chamber would work with your existing processes, or for the scalability of drug products that are moving out of R&D and into production, we've put together an ROI, or return on investment calculator, that allows companies to take a look at a number of different options, maybe their present options and some alternatives to those options, and determine really what it means from the bottom line, the investment, and the total amount of money that it is going to cost them to operate that process. So this tool can be tailored to specific processes, whether it's a customer who is expanding a production facility or who is changing the scale of a drug manufacturing operation. In that, we compare those different solutions and figure out how it's going to scale up. So now we're going to take a look at the inputs and outputs to that calculator. So, of course, some assumptions always have to go into a model. So what are those assumptions, and what do we feed the model so that we get the right outputs from it? First thing that we can put in is obviously the consumables, and this can be a range of consumables, whether they're Entegris or they're competitive solutions. We can put in cost models associated with all of those different consumable types. That includes bags, bottles, shells, tube sets, manifolds, anything that the customer may be using as part of their drug manufacturing process. One of the key areas that we look at is specifically the failure rates and why the process might fail, whether that is contamination that might be caused by open top filling, or robustness issues that can occur with multi-layer films at low temperatures, or any other type of failure that would be caused during handling. We do have data on this from some customers that we've worked with in the past that we can estimate that. Otherwise, we can put in information directly from a customer's existing process to make sure that we're as accurate as possible. Next is equipment costs for the freezing and filling system. So what is the capital equipment that is required to go through that cold chain process? And costs and timelines associated with adopting a new solution. So an existing solution obviously would not have a ramp, but if you're transitioning to a different type of equipment or a different process, we need to keep in mind that it does not instantly start and that you need to accommodate that in your return on investment calculations. Next, any kind of testing that's required for it. And finally, operating costs that would be related to the people that are needed for the process, the energy or the infrastructure to continue the operation of it. So based on those inputs, let's take a case study and see how this can be applied. So here we're going to discuss a customer who currently has a process that is using a combination of bottles and bags. So they have some 1 liter and 2 liter bottles, and then they have some bags in 6 liter, 12 liter, and 4 liter sizes. So this customer does have some existing infrastructure that we can use to evaluate that cost model. That's a combination of lab freezers, cold walls, as well as some from FARRAR. This customer is looking at creating 2 180 liter intermediate batches. That results in a 20 gram per liter concentration. And from a cost perspective, that means that you end up with about $50,000 per liter in total value of the product that needs to be frozen and stored. This customer does a large number of frozen shipments per year, meaning that they relocate that product from the manufacturing facility to a location where final fill is done. In this case, this manufacturer has 1 location where drug product is manufactured, but 5 different drug receiving sites that need to be taken into account because of the thaw portion of the process where those drug products would then be thawed. Customer wants 40 hours or less to thaw 35 liters. And then they've also given us a brief list of concerns or pain points that they have with their current process that they would like us to address. So here's an example of an output, the total cost over time that would come out of that ROI model. Here, we've already looked and seen that using the FARRAR controlled rate chambers as process freezers, meaning taking the drug product from ambient temperature to the low temperature that's required for transport, as well as using those controlled rate chambers for the thaw process, is the most economical method of capital that the customer should invest in here. And what's shown specifically is how a number of different consumables that are available in the market would react to that capital equipment selection that this customer has made. So here, starting on the left with 0 years going over to 4, you can see the total amount of cost over time. Compared to the customer's existing solutions, there's about a 1.25 year sort of crossover point where the customer would break even by making investments in changing to a different consumable set. And then over that 4-year time period, that 5 liter blast shell solution from Integris becomes the lowest cost consumable in the process, combined with those FARRAR controlled rate chambers. So overall, the Integris 5 liter blast shell in the FARRAR CRT provides the best value over this time interval for this particular customer application. So the recommendations and conclusions here would be to transition that product to the 5 liter blast shells from Integris. That would be 36 containers in the 5 liter size for each of their batch operations, storing a full batch in 4 freezers, and taking advantage of the Aramis film with secondary shell to reduce contaminant loss, as well as improve on the durability and reduce the breakage loss in that process. On the thawing side, the side that is often overlooked, here the FARRAR is also the best solution. It meets the thaw requirements with about 50 liters of capacity per batch, and can thaw 180 liters of that drug product in 60 hours. So that results in consistent thawing, a reproducible process. So this is a good example, but let's move on to some testing data that shows a little bit more of real-world applications.
So that ROI calculator, its ability to be customized for each unique freezing process, really helps remove the uncertainties around material and vessel selection. Rob's going to go into more detail here on 3 bag freezing studies, a 2 liter, 5 liter, and a 10 liter, and how they froze in different air style freezing options, ULT and a controlled blast freezer. He's also going to discuss the thawing process using the thaw cycle in the Model 4000. Finally, he'll give you some photos on how the bags fit into the Model 4000 CRC, so you can get a feel for what's going on.
So before we dive into the details of the blast shells, one of the things that got left out is how he worked with my team over the past 2 years to pick the right controlled rate chamber for our application. So by looking at the size of our facility, our HVAC concerns, our access to chilling systems, he and his team helped us pick the right controlled rate chamber to work with the blast shells that we had under development, and ultimately arrived on one of the liquid cooled controlled rate chambers to do the testing that you're about to see. So some basic specs of the blast shells that were used in the studies that were conducted. We had 2-liter, 5-liter, and 10-liter shells. Those 2-liter and 5-liter shells currently available. 10-liter still currently in prototype status, but is able to move forward with enough customer input. All of these shells are designed to work at both maximum fill, so rated fill is the size, so 2-liter rated fill in a 2-liter shell, but also all the way down to partial fills of 50%. So in that 2-liter shell, filling down to 1 liter is also acceptable and has been validated as part of our process. That gives a usable range of 1 liter to 2 liter in the 2-liter size, 2.5 to 5 in the 5-liter, and 5 liter to 10 liter in that largest size. Dimensions on the right-hand side are available to you if you're interested to see how these different size shells would work into the manufacturing process or the labs that you currently have as your facility. So the next part of the study was determining how many of these can be fit into different sized freezers. So we did studies using 4 different cold wall freezers and the FARRAR controlled rate chamber to see what that maximum fill volume would be with each of those sizes. Table is shown over here on the right. So with the FARRAR, 2-liter about 18 liters, 45 liters with the 5-liter, and about 100 liters with that 10-liter size, which compares well to some of the controlled rate chambers, of course, until you look at some of the data that represents how long it takes to freeze. So let's look at the data from freezing in these different types of freezers. The chart here shows freeze rates from the 2 and the 5-liter shell sizes. So the different colors represent the number of shells that were frozen in each of those freezers. The blue representing 1 shell, so a very light load in each of the freezers. The orange is 5 shells, so an intermediate, maybe close to a half load. And then the gray is the maximum capacity with the number of liters of each of those fills shown inside of the graph for each of those. What you'll note here is that there is a very large difference in the freeze time for each of these different freezer types. For our freezer on the left-hand side, significantly faster freeze rates, as well as being much more consistent between the different fill volumes that are located inside of that freezer. So overall, a much higher freeze rate to match that footprint data that Scott showed us earlier in the presentation. So overall, much faster freeze rates are achieved in the FARRAR controlled rate chamber than any of the cold wall freezer options tested. So if we start to look at the large blast freeze performance, we've also compared against a competitive product on market. So here our 10-liter blast shell was compared against a 12-liter from a competitive manufacturer. The solid lines in the chart represent the different freeze rates that we've got. So in the red, we've got the Integris, and then in the other color, we've got that competitive shell assembly. So what you'll notice here is that the Integris shells freeze faster by almost 5 hours. That's 35% faster than the competitor, even though the fill volume inside the freezer was almost identical in this case. So full freezer load, 35% faster only by changing the consumable type inside of that controlled rate chamber. And what you'll see is that the slowest freezing Integris shell was actually still faster than the competitor's fastest solution. You'll also note that there is a lower variance in fastest to slowest in the Integris shells than that of the competitive product. So we do get lots of questions about how our products scale. So what if we have a customer that has a process that needs a small consumable set to start with, but will ultimately evolve into a larger one? Here's an overlay that shows the freeze times of the 2-liter, 5-liter, and 10-liter all in one slide. 2-liter freezing fastest does have the lowest thermal mass in the freezer when it's got a full load installed. but very close performance between the 5-liter and 10-liter shells that demonstrates both the freezing capacity, so the power that that controlled rate chamber has, but also highlights some very interesting design changes that we made to the 10-liter shells to enhance its heat transfer that are now incorporated into that 10-liter shell design. So often overlooked, we'll transition into a couple of different methods of thawing and show some thawing data for our 5-liter shells. 4 different methods were tested here. So we had blast shells that were closed, just sitting on a bench, the cheapest and slowest, unfortunately, option in this case. We also had some options that they were on a shelf with a fan, also a half shell where a fan was applied to it, and lastly, thawing inside of the FARRAR controlled rate chamber. So the lines going horizontally from 0 to 21, you'll see that green line breaking out of the top of that chart at about 10 hours in time there. That's that FARRAR controlled rate chamber, bringing those shells up to 20 C or ambient temperature in approximately 10 hours or so on that chart. Whereas the other 3 options, you'll see continuing past 21 hours in time have still not yet reached that 20 degrees C. So the last slide we're going to show here is just a complete freeze thaw cycle. That's something that we can do inside of that controlled rate chamber. So starting with the drug products at an elevated temperature, freezing them all the way down to -80, allowing dwell, and then executing a thaw profile that brings them up to the temperature. So you do see a little bit of variation in those shell temperatures, generally caused by position inside the freezer, but overall a very tight spread of overall variance when freezing and thawing at maximum capacity inside that controlled rate chamber. So the last slide that we've got here, we do get questions about how our bags are sized in comparison to the shelving inside the controlled rate chamber. So while we generally don't advocate doing bare bags inside the freezer because of survivability during handling and the opportunity for damaging the bags in the frozen state, this is just an image that does show some of the different bag offerings that we have in our standard catalog for the Aramis bags, and showing how they would fit inside the freezer relative to the footprint of each of those shelves. So here, we'll transition back to Scott and talk about how we've solved some of those problems for our customers.
Thanks, Rob. That was great information. It really allows users to make an informed decision about which solution best fits their processes. Now, remember this slide from earlier. This time, we've got the solutions in green summarized so that we can revisit quickly how we did solving those most common workflow challenges. So from the financial bucket, the upfront capital investment, that FARRAR Model 4000 provides high flexibility, as you saw from the studies that we just showed, and costs a fraction of a plate freezer and provides much more utility, speed, and reliability than a ULT, while also taking up less space on the manufacturing floor. The cost to run have been addressed, of course, by the Model 4000 because its limited units are needed to run the process because of its speed. And the ability to use an air-cooled system means you won't have to install a chilled water line to cool the compressors. Finally, the excess cooling capacity, remember that 1616 watts of available capacity provides plenty of cooling power without stressing the compressors, leading to low maintenance and high reliability. The easy-to-use HMI also makes standing up lines fast and replicating them in other locations very easy. From the process and product challenges, we began with the vessel like we talked about, and we talked about the challenges with bottles. The Aramis bag materials are made with high purity materials and equipped with 3 lines for closed system processing, minimizing contamination. Both the FARRAR Model 4000 and the Integris blast shells significantly simplify the processing by allowing draining and filling in multiple orientations and configurations, and the bag's airflow promotion design increases the freezing speed when paired with a 4-stair blast freezer like FARRAR. These faster freezing times also reduce cryo concentrations, where the water in the solution freezes first, allowing the non-water drug substance material to concentrate and possibly become damaged, as well as increase processing speed through the freezing step. From the sustainability bucket, those uses of regulated chemicals by not using silicone oils and hazardous refrigerants in the freezer are making it safer for the environment when you're using the FARRAR blast freezer. In addition, the Integris blast shells have been qualified and validated for 3 freeze and thaw cycles. And when it's time to dispose of them, the materials are fully recyclable. No thin films or added effort to get the shells recycled. Single use here does not have to mean one use. So if after hearing us talk, you still have questions, we have ways to help you. First, you can ask them questions here in this webinar in a few minutes. And second, Rob and I will take just a few more minutes to talk about ways our teams can help guide you to your best fit solution without buying anything or without any obligations to buy anything.
So in addition to the Integris applications and commercial teams that are always available to assist with any questions, we also have available to us our Life Sciences Technology Center here in Billerica, Massachusetts. So this full service facility offers the opportunity to provide additional services for customers, whether it would be things like freezing system trials, evaluations of process, consumable testing, or all the way up to things like robustness testing using our drop tester or things like that on site. For additional information, we do offer tours of our facility. There are in-person tours where you can come to our location, but we also do have virtual options where we can show off the equipment and the processes that we've got on site. That includes a complete range of different freezing technologies, plate, blast, cold wall, as well as thaw systems, technologies that are unique to cell and gene therapy, pumping systems, liquid nitrogen-based operations, construction, and finally, information and technology available to capture data. So data acquisition systems, high-speed cameras, as well as other systems to capture data regarding products during the process.
FARRAR manufactures unique solutions that differ in looks and function to typical refrigeration equipment on the market. So we know you're going to have questions and concerns about placing materials in your process, materials and equipment in your process that you're unfamiliar with. You've heard Rob talk about services for running process validation studies in Integris's Tech Center. If you'd rather run the study on your own facility, we've got a program to make that happen too. So our Try Before You Buy demo program, it really is a hassle-free way to test drive a 4000 series rate chamber or a ULC with no obligations. This 8-week trial, or a little bit longer if that's what you need, gives you plenty of time to set up and run a number of tests. We start by discussing your facility with you, allowing you to prep the space to accommodate the unit before we get on site. We'll then deliver, install, and provide your team with a detailed training and best practices with the equipment. So if you also like help in designing a test protocol, we can help optimize the freezer settings for best results with our included tech support engineering consulting. Our equipment experts can even work with you on designing ideal material handling solutions that will increase your efficiency in the process. It's really simple to get started. You can go to our website and contact us with the subject Try Before You Buy demo program, and we'll get back to you within 3 business days or sooner. We'll walk you through the rest of the process, and it really is about as easy as it gets with capital equipment. All right, so Rob and I really would like to thank you for your time, and I hope you learned a great deal about ways to solve common freeze and thaw issues. We'll now open it up for questions you may have. Just type them in the chat window on your screen.
Many thanks to all the attendees from the Integris side, and looking forward to answering your questions.
Thank you, Rob and Scott. The question we have here, what factors would you consider in choosing an air or water-cooled controlled rate chamber for my operation?
I'll start with that, but I know Rob's got a firsthand experience with this. So what we start when we're thinking about which model to go after, air or water cooled, we usually start with the cooling capacity of the HVAC in the area. So if you've got a large warehouse, more than likely that's perfectly fine for an air cooled unit. If you've got a small area that has to be precisely cooled for the rest of your process, you might have to upsize your HVAC unit if there's a lot of heat coming around. That might make it easier for a water cooled if you have a chiller and you have chilled water in the area anyway. But there's a couple of different factors that our team of engineers can work with you to determine the best for your space and the best cost. But I'll let Rob make a couple comments on what he's experienced in the tech center.
Sure. So that was really one of the first questions that we had when we saw the different offerings in the 4000 series. And we ended up walking through some of the architectural plans that we had for our Life Sciences Technology Center. What we found is that we were taking over a space that was previously occupied by another lab. And even though we had good air exchange, when we did the math and the calculations for heat load on one of the air-cooled chillers, it really would have overtaken some of the HVAC systems that were currently installed in the space. But in doing the math, we found that we actually had excess capacity in one of the liquid chilling systems in the building. That allowed us to transition to one of the liquid cooled systems to better match the needs of our facility as a whole.
Another question here, are studies ongoing using liquid nitrogen or other more environmental friendly cooling media?
Rob, you want to take that one?
Sure. So on the Integrist side, we do a lot of processes that do use liquid nitrogen. We've done example studies for customers using everything from direct immersion to systems that use gas, so the gas phases of nitrogen for the cooling process. Integrist does not sell any systems that currently are actually freezer systems. We focused exclusively on the consumable side of that, but we're happy to operate any studies of any of our shell systems all the way down to cassettes. Some aluminum cassettes were shown. They're actually a little bit smaller than our blast shells. We're happy to tailor any testing to a liquid or gaseous nitrogen system using those containment systems as well as our Aramis bag assemblies.
Another question here, I'm not sure how this would relate to blood collection facility and freezing plasma in collection bags. Can it be scaled to that?
I'll take that one, Rob, to start. You might have some comments based on what I say, but this is a great question. In fact, we have been looking at this as a way to give you an alternate solution to like a thermogenesis freezing solution for plasma. In this particular case, the rate chamber could easily be scaled to freeze down to the temperature you want, typically around -30. We could do that, or even could be -35. That's not a problem. I think that modifying your protocol to be able to add materials on a less regular basis. I think what I've seen with the thermogenesis units have been that you might add a batch of plasma to be frozen every 20 to 30 minutes if you're in a high volume process area. This probably would require you to let it run for a couple of hours before moving the material out to a freezer. Alternatively to that, we also have walk-in solution offerings that we do design specifically for plasma freezing and storage. So if you're interested in that, we can also talk about that. But there is a method in which you can use the rate chamber for this.
I think you covered that great, Scott. Nothing to add on my side.
Okay.
Next question here, can you help companies develop freezing protocols? Is freezing faster always better unless you are freezing cells and want to use a 1 degree Celsius minimum cooling rate?
I think both of us probably have an opinion on this one. I'll let you start, Rob.
Sure. So in the Integris, the Life Sciences Technology Center, we obviously have controlled rate freezers. What we can do is we can work with customers as long as we don't have virulent biologics inside of that tech center. So we do need to keep in mind that it's generally a clean facility, but we can work with customers to determine what the correct freeze rate for that is. Some of that comes down to selection of the consumables. Scott talked a little bit earlier about the relationship between surface area, volume, and conductivity of materials. There are some consumables that may not reach some of the rates that our customers are looking for, and there are others that might. And the rest is the combination of using the specific recipes inside of those controlled rate freezing systems to better tailor to what the customer is looking for. So maybe add a little bit more on that side, Scott.
Yeah, so what we've seen also is that we can definitely help with the freezing protocols, and is freezing faster always better? For the most part, we've seen the answer to that is yes, especially as it pertains to cryo concentrations. So what that entails, it requires some testing and some really an understanding of the crystal structure that forms and how it forms. So I think without knowing exactly the process and exactly what might be frozen, I'd say on average the answer is yes, freezing faster is always better.
Another question here. We have a requirement to provide specific product freeze rates between 0 degrees Celsius and -10 degrees Celsius. Is this possible with the system discussed?
It is possible. We have to set up a specific program to do that inside the controller of the rate chamber, and we can start the process and have it end at between 0 and -10. Well, it would be a pretty fast process, but definitely can be done.
Another question. Ergonomics are a major concern at our facility. How do your solutions address handling risks?
So I'll start with that from the consumable side, Scott.
Mm-hmm.
So if you look at the designs of some of our shells and some of the competitive products in the blast freezing space are sort of really smooth metal shells. And the problem with that is if you have condensation on the outside, you get ice layers, it can become very slippery and it can be difficult to handle using cryogenic gloves. So we've designed those special handle assemblies into our Integris blast shells, designed to either have a complete grip around the outside if you've got larger hands, maybe don't have gloves on, and then use some of the inner hand holds in there to ensure that you get a good grip on that product. In addition to that, the polymer frames of those shells are relatively light. So if you're looking at total mass carried, they tend to be lighter than some of those metal solutions that are on the market. So if you're bumping into a limit of, let's just say, total mass carried or total mass transported through your facilities, we've got a pretty light solution there in that 2 and that 5 liter size. So by offering that 2 liter, that 5 liter, and potentially that 10 liter, we can work within the ergonomic constraints that you'd have within your facility, or look at the points that it might be relevant to transition to handling systems to move some of these items through your workflow, as opposed to having them moved manually.
And from the equipment handling side, we have a specialized team at FARRAR that can work with you to design a specific material handling process and equipment. It really has been a solid way that we interact and making processes more streamlined. So we can look at what moving equipment you might have. If it's human moving, then we understand how to develop certain carts that would help transition materials between freezers. We also, and processes, or if you have machine, we also can design ones that work with the types of machines you have. We do have some that we offer now in more common cart sizes and cart dimensions that would take some of the larger pieces of vessels like 10 liter and 20 liter bags and bottles, and we can always modify for other sizes and shapes as well. So it's pretty wide open in how we can do it, but we're usually very fast and very in tune with asking the right questions for your process to make it smooth.
Thank you, everyone. Great questions. We appreciate you joining us today and sticking with us as we are over time. A brief survey will launch following the webinar, and any feedback is greatly appreciated. A recording of this presentation will be available on our website by the middle of next week. As a reminder, links to past webinars can be found on our website at FARRARscientific.com, as well as our YouTube page, and we hope that you would join us in a future webinar. Thank you and have a great day.